Hello everyone, this is Professor Beda. Let's talk about meiosis and sexual life cycles. Living organisms are best distinguished by their ability to reproduce their own kind, the transmission of traits from one generation to the next is called inheritance or heredity. At the same time, sons and daughters are not identical copies of their parents or of their siblings. Along with inherited similarity, there is also variation. The study of both heredity and inherited variation is called genetics. Parents endow their offspring with coded information in the form of heredity units called genes. Our genes program specific traits that emerge as we develop from fertilized eggs to adults. The genetic program is written in the language of DNA, the polymer of 4 different nucleotides, and inherited information is passed on in the form of each gene's specific sequence of DNA nucleotides. Most genes program cells to synthesize specific enzymes and other proteins, whose cumulative action produces an organism's inherited traits. The transmission of hereditary traits has its molecular basis in the replication of DNA, which produces copies of genes that can be passed from parents to offspring. In animals and plants, reproductive cells called gametes are the vehicles that transmit genes from one generation to the next. During fertilization, male and female gametes, spring and eggs, unite, passing on genes of both parents to their offspring. Every species has a characteristic number of chromosomes. For example, humans have 46 chromosomes in their somatic cells. Um, you know that somatic cells, all cells of body except the gametes and their precursors. Each chromosome consists of a single long DNA molecules elaborately coiled in association with various proteins. We call them histone proteins. One chromosome includes several hundreds to few thousands of genes, each gene being a precise sequence of nucleotides along the DNA nucleotide. A gene's specific location along the length of chromosome is called the gene's locus or loci. Only organisms that reproduce asexually have offspring that are exact genetic copies of themselves. In asexual reproduction, a single individual, like yeast or an amoeba, is the sole parent and passes copies of all its genes to its offspring without the fusion of gametes. For example, single-celled eukaryotic organism can reproduce sexually by mitotic cell division, in which DNA is copied and allocated equally to two daughter cells. The genomes of the offspring are virtually exact copies of the parent's genome. Some multicellular organisms are also capable of reproducing asexually, like Hydra and redwoods, because the cells of the offspring arise via mitosis. In the parent, the offspring is usually genetically identical to its parent. An individual that reproduces asexually gives rise to a clone, an individual or a group of individuals that are genetically identical to the parent. In sexual reproduction, Two parents gives rise to offspring that have unique combination of genes inherited from the two parents. In contrast to a clone, offspring of sexual reproduction vary genetically from their siblings and both parents. So genetic variation is an important consequence of sexual reproduction. A life cycle is the generation to generation sequence of stages in the reproductive history of an organism uh, from conception to production of its own offspring. A careful examination of micrograph of 46 human chromosomes from a single cell in mitosis reveals that there are two chromosomes of each of the 23 types. This becomes clear when images of chromosomes are arranged in pairs, starting with the longest chromosomes. The resulting ordered display is called karyotype. In human, each somatic cell has 46 chromosomes. Before mitosis begins, the chromosomes are duplicated. During mitosis, the chromosomes become condensed enough to be visible under light microscope. At this point, they can be distinguished from one another by their size, the position of their centromere, and the pattern of colored bands produced by a certain chromatin binding stain. So karyotype is a display of condensed chromosomes arranged in pairs. Karyotyping can be used to screen for defective chromosomes or abnormal number of chromosomes associated with certain congenital disorders, such as Down syndrome. Uh, Karyotypes are prepared from isolated somatic cells, which are treated with a drug to stimulate mitosis and then grown in culture for several days. Cells arrested when the chromosomes are most highly condensed and usually at metaphase, and then stained with special dyes, then viewed with microscope equipped with a digital camera. Uh, an image of chromosome is displayed on a computer monitor, and a digital software is used to arrange them in pairs according to their appearance. This karyotypes shows the chromosomes from a human male as seen by the presence of the YXY chromosome pair. Uh, the size of chromosome, position of chromosome, centromere, and pattern of stained bands help identify specific chromosomes. Although it's difficult to discern in the karyotype, each metaphase chromosome consists of two closely attached sister chromatids. Um, and if you see this diagram, the enlarged diagram, it's appeared that there is a pair of homologous duplicated chromosomes. The two chromosomes of a pair have the same length, centromere position, and staining patterns. These are called homologous chromosomes or homologs, and both chromosomes of each pair carry genes controlling the same inherited characters. Uh, for example, um, if we have uh, if there is a gene for eye color is situated at a particular locus on a certain chromosome, then its homologous chromosome uh, will also have a version of the eye color gene at equivalent locus. The two chromosomes referred to as X and Y are an important exception to the general pattern of homologous chromosomes in human somatic cells. Typically, human females have a homologous pair of X chromosome, XX, while males have one X and one Y chromosome. Only small part of the X and Y are homologous. Most of the genes carried on the X chromosome don't have counterpart on the tiny Y chromosome, and the Y chromosome has genes lacking on the X chromosome. Due to their role in sex determination, the X and Y chromosomes are called the sex chromosomes, while the other 22 pair of chromosomes are called autosomes. We inherit one chromosome of a pair from each parent. Thus, the 46 chromosome in our somatic cells are usually two sets of 23 chromosomes, a maternal set from our mother and paternal set from our father. The number of chromosomes in a single set is represented by N. Any cell with two chromosome set is called diploid cell and has a diploid number of chromosomes, abbreviated to N. For human, the diploid number is 46. That means the 2N is 46, the number of chromosomes in our somatic cells. In a cell in which DNA synthesis has occurred, all the chromosomes are duplicated, and therefore each consists of two identical sister chromatids associated closely at the centromere and along the arms. Even though the chromosomes are duplicated, we still say the cell is diploid or 2N. This is because it has only two sets of information regardless of the number of chromatids, which are merely copies of the information in one set. Unlike somatic cells, gametes contain a single set of chromosomes. Such cells are called haploid cells, and each has a haploid number of chromosomes, N. For human, the haploid number is 23, N equal to 23. The set of the 23 consists of 22 autosomes and a single sex chromosome. An unfertilized egg ovum, the sex chromosome is X, and in sperm, the sex chromosome may be either X or Y. Each sexually reproducing species has a characteristic number, diploid and haploid number. For example, fruit fly has a diploid number 2N of 8 and haploid number N of 4, while for dogs 2N is 78 and haploid number N is 39. A cell from an organism with a diploid number of 6 to n equals 6 is depicted here, following chromosome duplication and condensation. Each of the 6 duplicated chromosomes consists of 2 sister chromatids associated closely along their length. Each homologous chromosome or pair is composed of chromosome from maternal and the red one and one from paternal set, the blue. Each set is made up of three chromosomes. In this example, we have a long, medium, and short. Together, one maternal and one paternal chromatid is a pair of, or in a pair of homologous chromosomes are called non-sister chromatids. The human life cycle begins when a haploid sperm from the father fuses with a haploid egg from the mother. This union of gametes culminating in fusion of their nuclei is called fertilization. The resulting fertilized egg or zygote is diploid because it contains two haploid sets of chromosomes bearing genes representing the maternal and paternal family lines. As a human develops into a sexually mature adult, mitosis of the zygote and its descendant cells uh, generates all the somatic cells of the body. Both chromosomes sit in the zygote and all the genes they carry are passed with precision to the somatic cell. The only cells of the human body not produced by mitosis are the gametes, which develop from specialized cells called germ cells in the gonad, ovaries in females, and testes in males. Um, Imagine what would happen if human gametes were made by mitosis. They would be diploid like the somatic cells. At the next round of fertilization, when two gametes fused, the normal chromosome number of 46 would double to 92, and each subsequent generation would double the number of chromosomes yet again. Uh, so mitosis in gametes will not happen. Of course, this doesn't happen because in sexually reproducing organisms, gamete formation involves a type of cell division called meiosis. This type of cell division reduces the number of sets of chromosomes from two in the parent cell to one in each gamete, uh, counterbalancing the doubling that occurs at fertilization. As a result of meiosis, each human sperm and egg is haploid, n equal 23. Fertilization restores the diploid condition by combining two sets of chromosomes, and the human life cycle is repeated generation after generation. Although the alternation of meiosis and fertilization is common to all organisms that reproduce sexually, the timing of these two events in the life cycle varies depending on the species. These variations can be grouped into three main types of life cycles. In animals, in the type that occur in human and most of other animals, gametes are only haploid cells. Meiosis occurs in germ cells during the production of gametes, which undergo no further cell division prior to fertilization. After fertilization, the haploid zygote divides by mitosis, producing a multicellular organism that is diploid. Sorry, the zygote is diploid. So after fertilization, we have a diploid zygote. Then zygote divides by mitosis, producing multicellular organism that is diploid. In plant and some species of algae exhibit a second type of life cycle called alternation of generation. This type includes both diploids and haploid stages that are multicellular. The multicellular diploid stage is called sporophytes. Uh, Meiosis in the sporophytes produces haploid uh, cells called spores. Unlike gametes, a doesn't haploid spore doesn't fuse with another cell but divides. Divides, of course, mitotically, generating a multicellular haploid stage called gametophyte. Uh, Cells of the gametophyte give rise to gametes by mitosis. Fusion of two haploid gametes as fertilization results in a diploid zygote, which develops into the next sporophyte generation. Therefore, in this type of life cycle, the sporophyte generation produces gametophyte as its offspring, and the gametophyte generation produces sporophytes. A third type of life cycle occurs in most fungi and some protists, including some algae. After gametes fuse and form a diploid zygote, meiosis occurs without a multicellular diploid offspring developing. Meiosis produces not gametes but haploid cells that then divide by mitosis and give rise to either unicellular descendants or haploid multicellular adult organism. Subsequently, the haploid organism carries out further mitosis, producing the cell that develop into gametes. The only diploid stage found in these species is the single-celled zygote. Uh, Note that either haploid or diploid cells can divide by mitosis, depending on the type of life cycle. Only diploid cells can undergo meiosis. Haploid cells cannot because they already have a single set of chromosomes that cannot be further reduced. Uh, Through the three types of sexual life cycle uh, differs in the timing of meiosis and fertilization, they share fundamental results, genetic variation among offspring. Several steps of meiosis closely resemble corresponding steps in mitosis. Meiosis, like mitosis, is preceded by an interphase, which includes the S phase, the duplication of chromosome. Uh, However, this is followed by not one but two consecutive cell division called meiosis 1 and meiosis 2. These two division results in four daughter cells rather than the two uh, daughter cells of mitosis, each with only half as many chromosomes as the parent cell, one set rather than two. In this overview of meiosis shows for a single pair of homologous chromosomes in a diploid cell that both members of the pair are duplicated and the copies sorted into four haploid daughter cells. Recall that sister chromatids are two copies of one chromosome, closely associated all along their length. This association is called sister chromatid cohesion. Together, the sister chromatids make up one duplicated chromosome. In contrast, the two chromosomes of homologous pair are individual chromosomes that were inherited from each parent. Homologous appear alike in the microscope, but they may have different versions of genes at corresponding loci. Each version is called an allele of that gene. For example, one chromosome might have an allele for freckles, but the homologous chromosome may have an allele for the absence of freckles at the same time or at the same locus. This figure describes in detail the stages of the two division of meiosis, meiosis I and meiosis II, for an animal cell whose diploid number is 6. Let's start with the first stage or the first division of meiosis I. The first stage is separation of homologous chromosomes. So at Prophase I, centrosome movement, uh, spindle formation, and nuclear envelope breakdown occurs as in mitosis. Chromosomes condense progressively throughout Prophase I. During early Prophase I, before the stage shown here, each chromosome pairs with its homologue, um, aligned gene by gene, and crossing over occurs. The DNA molecule of non-sister chromatids are broken by proteins and are rejoined to each other. At this stage, each homologous pair has one or more X-shaped region called chiasmata, singular chiasma, where crossover have occurred. Later in Prophase I, microtubules from one pole or the other attached to the kinetochores, one at the centromere of each homologue. The two kinetochores on the sister chromatids of homologous link together by proteins uh, and act as a single kinetochore. Microtubules move the homologous pairs toward the metaphase plate. Prophase I of meiosis is a very busy time and considered very important molecular basis for inherited variation. After interphase, the chromosomes have been duplicated and sister chromatids are held together by proteins called cohesins. Cohesins, those the purple rings. Each pair of homologous associated along their length. Uh, the DNA molecule of the two non-sister chromatids are broken at precisely corresponding points. Um, the second stage, a zipper-like protein complex, the synaptonemal complex, the green, begins to form, attaching one homologue to the other. the chromatin continues to condense. The synaptinimal complex is fully formed. Uh, the two homologs are said to be synapses. During synapsis, the DNA breaks are closed up when each broken and is joined to the corresponding segment of non-sister chromatid, producing crossover. After the synaptinimal complex disassembles, the homologs move slightly apart from each other but remain attached because of sister chromatid cohesion. Even though some of the DNA may no longer be attached to its original chromosome, the points of attached to its original chromosome, the points of attachment where crossover have occurred, show up at chiasmata. The chromosomes continue to condense as they move toward the metaphase plate. In Metaphase I, of pairs of homologous chromosomes are now arranged at the metaphase plate with one chromosome of each pair facing each pole. Each pair has lined up independently of other pairs. This arrangement is called independent assortment. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. The chromatids of the other homolog are attached to microtubules from the opposite pole. An anaphase breaking down of proteins that are responsible for sister chromatid cohesion along chromatid arms allows homologs to separate. The homologs move toward the opposite poles, guided by the spindle apparatus. Sister chromatid cohesion persists at the centromere, causing the two chromatids of each chromosome to move as a unit toward the same pole. When telophase I begins, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of two sister chromatids. One or both chromatids include a region of non-sister chromatid DNA. Cytokinesis, division of the cytoplasm, usually occurs simultaneously with telophase I, forming two haploid daughter cells. In animal cells like these, a cleavage furrow forms, and remember that in plant cells, a cell plate forms. Um, in some species, chromosomes condense and nuclear envelope form. No chromosome duplication occurs between meiosis I and meiosis II. So keep in mind that no chromosome replication occurs between the end of meiosis I and the beginning of meiosis II because the chromosomes are already replicated. Um, in meiosis II is very similar to mitosis. And let's start with prophase. So here we have prophase II, a spindle apparatus form. And in late prophase II, chromosomes each is still composed of two chromatids associated at the centromere, are moved by microtubules toward the metaphase II plate. The chromosomes are positioned at metaphase plate as in mitosis in the second stage of mitosis in metaphase II. And because of crossing over in meiosis I, the two sister chromatids of each chromosome are not genetically identical. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles. The third stage is Anaphase II, a breakdown of proteins holding the sister chromatids together as the centromere allows the chromatids to separate and move towards opposite poles. Each chromatid has now become an individual chromosome. Telophase II and cytokinesis. In telophase II, just like mitosis, nuclei form and the chromosomes begin condensing, decondensing, and cytokinesis occurs. The meiosis division of one parent cell produces four cells, daughter cells, each with a haploid set of unduplicated chromosomes. The four daughter cells are genetically distinct from one another and from the parent cell. In a comparison of mitosis and meiosis, we'll see that meiosis produces four cells and reduces the number of chromosome sets from two diploid to one haploid, whereas mitosis produces two cells and conserves the numbers of chromosome sets. Meiosis produces cells that differ genetically from the parent cell and from each other, whereas mitosis produces daughter cells that are genetically identical to their parent cell and to each other. This figure summarizes the key differences between meiosis and mitosis in diploid cells. DNA duplication or replication. In mitosis, the DNA duplication occurs during interphase before mitosis begins. Whereas in meiosis, DNA duplication occurs during interphase before meiosis I, but not before meiosis II. Number of division in mitosis, including prophase, prometaphase, metaphase, anaphase, and telophase. In meiosis, two divisions occur, each including prophase, metaphase, anaphase, and telophase. If we want to compare synapses of homologous chromosomes, in mitosis doesn't occur. So in mitosis, there is no synapsis of homologous chromosomes. Meiosis occurs during prophase I and along with the crossing over between non-sister chromatids, resulting chiasmata hold pairs together due to sister chromatid cohesion. Number of daughter cells and generic composition. In mitosis 2, we have two daughter cells, each genetically identical to parent cell with the same number of chromosomes. In meiosis 4, we have four daughter cells, each haploid N, genetically different from the parent and from each other. Uh, the fifth difference is role in animals, fungi, and plants. In mitosis, enable multicellular animal like fungus or plants, gametophyte or sporophyte, to arise from a single cell, produces cells for growth, repair, and in some species, asexual reproduction produces gametes in plant gametophyte. In meiosis, produces gametes in animals or spores in fungi and in plant sporophytes, reduces number of chromosome sets by half, and introduces genetic variability among the gametes or spores. Uh, so three events unique to meiosis occur during meiosis 1, synapsis and crossing over during prophase 1, duplicated homologs pair and crossing over occur. Synapses and crossing over don't occur during prophase of mitosis. Alignment of homologous pairs at the metaphase plate. So at metaphase plate or metaphase at the metaphase 1, sorry, of meiosis, pairs of homologous are positioned at metaphase plate rather than individual chromosomes as in metaphase of mitosis. The separation of homologous at anaphase 1 of meiosis, the duplicated chromosomes of each homologous pair move toward opposite poles, but the sister chromatids of each duplicated chromosome remain attached. In anaphase of mitosis, by contrast, sister chromatids separate. So how do we account for genetic variation of the family members? Mutations are the original source of genetic diversity. These changes in an organism's DNA create the differentiate or different versions of genes known as alleles. Once these differences arise, um, reshuffling of the alleles during sexual reproduction produces the variation that results in each member of a sexually reproducing population having a unique combination of traits. In species that reproduce sexually, the behavior of chromosomes during meiosis and fertilization is responsible for most of the variation that arises in each generation. Three mechanisms contribute to the genetic variation arising from sexual reproduction. We have independent assortment of chromosomes, crossing over, and random fertilization. Independent assortment of chromosomes, one aspect of sexual reproduction that generates genetic variation is the random orientation of pairs of homologous chromosomes at metaphase of meiosis 1. At metaphase 1, the homologous pairs, each consisting of one maternal and one paternal chromosome, are situated at the metaphase plate. Each pair may orient with either its maternal or paternal homologue closer to a given pole. Its orientation is as random as the flip of a coin. Thus, there is 50% chance that a particular daughter cell of meiosis 1 will get the maternal chromosome of a certain homologous pair and 50% chance that it will get the paternal chromosome. because each pair of homologous chromosome is positioned independently of the other pairs at metaphase I. The first meiotic division results in each pair sorting its maternal and paternal homologs into daughter cells independently of uh, every other pair. This is called independent assortment. Uh, each daughter cell represents one outcome of all possible combination of maternal and paternal chromosomes. As shown here, the number of combination possible for daughter cells formed by meiosis of a diploid cell with two pairs of homologous chromosome 2n is 4. Two possible arrangements for the first pair times two possible arrangements for the second pair. In general, the number of possible combinations when chromosomes sort independently during meiosis is 2 to the power n, where n is the haploid number of the species. In the case of humans, n is 23. The number of possible combinations of maternal and paternal chromosome in the resulting gametes is 2 to the power 23, or about 8.4 million. Each gamete that you produce in your lifetime contain one of roughly 8.4 million possible combinations of chromosomes. This is an underestimate because it doesn't take into account crossing over, which we'll consider next. Uh, in crossing over as the consequence of independent assortment of chromosomes during meiosis, each of us produces a collection of gametes differing greatly in their combinations of the chromosomes we inherited from our two parents. In previous figure suggests that each chromosome in a gamete is exclusively maternal or paternal in origin. In fact, this is not the case because uh, crossing over produces recombinant chromosomes, individual chromosomes that carry genes or DNA from two different parents. In meiosis, in humans, an average of 1 to 3 crossover events occur per chromosome uh, pair, depending on the size of chromosomes and the position of their chromosomes. So again, during meiosis that occur in human, an average of 1 to 3 crossover events occur per chromosome pair, depending on the size of chromosomes and the position of their centromere. Crossing over produces chromosomes with new combinations of maternal and paternal alleles. At metaphase II, chromosomes that contain one or more recombinant chromatids can be oriented into alternative non-equivalent ways with respect to other chromosomes because their sister chromatids are no longer identical. The different possible arrangements of non-identical sister chromatids during meiosis II further increase the number of generic types of daughter cells that can result from meiosis. So the important point is that crossing over by combining DNA inherited from two parents into a single chromosome, is an important source of genetic variation in sexual life cycle. Um, the random nature of fertilization adds to the genetic variation arising from meiosis. In human, each male and female gamete represent one of about 8.4 million. That means 2 to the power 23 possible chromosome combination due to independent assortment. The fusion of a male gamete with a female gamete during fertilization will produce a zygote with any of about 70 million. That means 2, uh, 2 to the power 23 by 2 to the power 23 diploid combinations. If we factor the variation brought about by crossing over, the number of possibilities is truly astronomical. Uh, you are really unique. Now that you've learned how new combinations of genes arise among offspring in a sexually reproducing population, how does the genetic variation in a population related to evolution? Darwin recognized that a population evolves through the differential reproductive sexes of its variance members. On average, those individuals best suited to the local environment leave the most offspring, thereby transmitting their genes. Thus, natural selection results in the accumulation of genetic variations favored in a certain environment. As the environment changes, the population may survive if in each generation at least some of its members can cope effectively with the new conditions. Mutations are the original source of different alleles, uh, which are the mixed and matched during meiosis. In a stable environment, sexual reproduction seems as it would be less advantageous than sexual reproduction, which ensures perpetuation of sexual combination of alleles. So, the ability of sexual reproduction to generate genetic diversity is the most commonly proposed explanation for the evolutionary persistence of this process.